Hello students. So in this video, let's do biochemistry snapshot images. As you know, these days many image based questions are asked. So you need to practice your image based questions in this subject also. Although till date not many image based questions have been asked in the exam, but still, so it can be asked, that's why we have to do it, right? So this video is very important to watch. So let's get started. First we will do the biochemical test. So let's see some of the test tubes and the beautiful colors they are giving. So let's do all the purple color test on this page so that you don't get confused regarding the colors. So we'll do all the purple colored positive reactions here. So first is Molish test. Molish test is for carbohydrates. It is a general test for all the carbohydrates, but there is one condition, that number of carbons should be 5 or more. So a 3 carbon or 4 carbon carbohydrate will not give Molish test positive, right? So what is the positive test here? The positive test is a purple colored ring at the junction of the two liquids, right? So this was Molish test. Then is Rothera's test. Rothera's test is for ketone bodies. There also we have purple colored ring. Okay. So if this image is given in the question, purple colored ring, then you really need to take care that the question is leading you towards carbohydrates or ketone body, right? So you have to be careful here. Then there is purple ring in one more test, that is for amino acid. In the color reactions of amino acids, that is Hopkin test. Hopkin test also gives a purple ring at the junction of two liquids, and this test, Hopkin test, is for which amino acid? It is for the aromatic amino acid tryptophan. Okay. Then we have Biuret test and ninhydrin test, where this is a purple colored solution which is a positive test. Okay. So be careful, there is purple ring in Molish, Biuret and Hopkin-Coles test, but there is purple color solution in Biuret and ninhydrin test. Let's quickly revise. Ninhydrin test is for what? It is for alpha amino acids, right? But this is not positive for proline and hydroxyproline. Okay. Then Biuret test is also for proteins and peptides. For proteins and peptides, a free amino acid or a dipeptide will not give Biuret test positive, because in Biuret test minimum 2 peptide bond compound should be there to give this test positive, and 2 peptide bonds are present in tripeptide and other higher proteins and peptides. Okay. So bi means 2, and here it refers to 2 peptide bonds. Okay. So you should watch all these basic videos in the previous, all the videos which I have taken. You should watch the videos on color reaction of proteins and amino acids, right? So here I am just taking all the images quickly. Otherwise I have covered everything in the topic videos. So ninhydrin test. Ninhydrin test is used for obtaining fingerprints, right? There's a ninhydrin spray which is available. We spray this on the person's fingers or hands, and then we can obtain a fingerprint. What happens? How ninhydrin reaction is occurring here? The ninhydrin spray contains ninhydrin reagent, and in your hands or in your fingers there is sweat. Sweat has amino acid. So amino acid, free amino acid, will react with this ninhydrin reagent to give this purple color, and that can be used to obtain fingerprints, right? Then, so one uses fingerprints. Other use of ninhydrin test is in paper chromatography. Please watch the video on chromatography also. So What happens, or the principle of paper chromatography, I have clearly explained in that video. So here you can just look at this image, that a paper is there or a thin layer of silica can be there, and you can see there are purple stains. So what happens, we apply amino acids here, and during this chromatography, amino acids, they move upward, and various interpretations we can conclude, right? So because we can see the amino acid using the ninhydrin reagent here, okay? Then I have taken all the red color tubes on a single page. It will be very, very easy for you to recall and remember. So first one is Millen's test. Millen's test for tyrosine. This is for tyrosine, okay? Tyrosine is an aromatic amino acid. Then Paul's test or Pauli's test. These are all color reaction of amino acids, right? This Pauli's test also gives red color. Millen's test also gives red color. Sakaguchi test also gives red color, but they are positive for different compounds. This Pauli's test is positive for tyrosine and histidine. Okay, tyrosine and histidine gives Pauli's test positive. Okay, then Sakaguchi test, which also gives red color, that is for arginine, which is a basic amino acid, right? Then Benedict's test. Benedict's test is given by reducing sugars. It is given positive by reducing sugars. Which are reducing sugars? All the monosaccharides are reducing in nature, like glucose, galactose, fructose, right? All are monosaccharides. Then some disaccharides are reducing in nature. For example, maltose, isomaltose and lactose. Okay, also learn the constituents of all these disaccharides, already covered in carbohydrate chemistry. Okay, so Benedict's test is also called a semi-quantitative test. It is semi-quantitative test because this test gives various colors. You can see in the picture, it gives various colors, and these colors tell the amount of sugar present. Amount means quantity. So it is qualitative test as it tells the presence of sugar or not, right? But it is semi-quantitative because it gives an estimate of the amount of sugar present in the sample. Okay, so see here, see the Benedict's reagent is blue in color. If after boiling the solution with the sample, blue remains blue, means negative, there is no sugar present. Okay, now otherwise there are many colors which can come: green, orange, yellow, brick red precipitates. So now how to learn here? You know the color spectrum Vibgyor. Let's write that Vibgyor in a sequence. Delete this V and I, we don't need that. Starting from B, that is B for blue, then G for green, then Y yellow, then O orange, then R red. Okay, so blue means negative. If green color comes in solution, then 1 plus. If yellow, then 2 plus. If orange, then 3 plus. This is the amount of sugar which is present. Red means 4 plus, right? So an idea is that sugar is increasing, the amount of sugar is increasing as the color is increasing in this color spectrum range. Okay, so red means very high amount of sugar is present. Okay, so this says traces of sugar, orange says moderate, and brick red precipitate says large amount of sugar present. Okay, so Benedict's test is qualitative as well as semi-quantitative. Okay, then there are many non-sugar substances which gives Benedict's test positive because these substances are reducing in nature, right? They are reducing in nature. So let's see these substances. These are uric acid, ascorbic acid. So uric acid, ascorbic acid, homogentisic acid, then drugs like salicylates, aspirin, glutathione, and creatinine. Okay, so learn this list. This is very, very important, that what are the non-sugar substances giving Benedict's test positive. Then coming to the next test, have you noticed in your first year of biochemistry practicals, when you were having your practical classes, then sometimes your hands were yellow in color? Hmm? And that was specially occurring when on some day you are handling nitric acid. So sometimes nitric acid gets, you touch nitric acid little bit on the bottle and your hands are yellow. So what was that? Let's see. So your hands turned yellow because of xanthoprotic reaction occurring, and this test gives yellow color, and this test is for aromatic amino acids. All the aromatic amino acids will give this test positive, but not phenylalanine. Okay, so again here in your hands there is sweat. In sweat, amino acids are there, aromatic amino acids. They will react with nitric acid, that is the reagent of xanthoprotic reaction, and that's why your hands turned yellow. Then next, coming to DNPH test, dinitrophenylhydrazine test, where the positive reaction is orange or yellow crystalline derivatives. So this is positive, this is negative. This test is done for the diagnosis of phenylketonuria and MSUD, maple syrup urine disease. This test detects alpha keto acids in urine, alpha keto acids. Now in phenylketonuria, which alpha keto acid comes in urine? That is phenyl pyruvate. Then in MSUD, there is alpha keto acids coming in urine. That's why there will be DNPH test positive, right? Then see the picture and tell me which is this biochemical method of blood glucose estimation. You know that routinely this is used at home. This is glucometer. So the question is, which biochemical test is happening here inside the glucometer? So that test is GOD, POD, enzymatic method. Enzymatic method of glucose estimation. GOD stands for glucose oxidase, POD stands for peroxidase, right? I have already covered the GOD POD test details in carbohydrates. Okay, so here we are just looking at the images. Then next, coming to CNT test, that is cyanide nitroprusside test. The positive test is purplish, or to be more precise, magenta color, and this test is positive for any substance containing sulfhydryl group or disulfide bond. For example, cysteine, cysteine, homocysteine, homocysteine. When we write cysteine, then we have disulfide bond. When we write cysteine or homocysteine, then there is sulfhydryl group. Okay. So in which diseases this test will be positive? It will be positive in homocysteinuria, cysteinuria, and cystinosis. Okay, cyanide nitroprusside test will be positive. Then next, coming to sulfur test. So positive sulfur test means it has sulfur containing amino acid, and the positive color is black or brown color. And this sulfur test, be careful, there are two sulfur containing amino acids, cysteine and methionine, right? Sulfur test will be positive for cysteine, but for methionine sulfur test is not positive, because in methionine, if you remember the structure of methionine, sulfur is attached to two carbons. Sulfur is hidden between two carbons. It is not exposed outside, right? So this sulfur cannot be easily released. So methionine will not give sulfur test, right? Next, coming to heat coagulation test. If you remember, you were doing it a lot in your first year biochemistry practicals, that you take protein solution and you fill 2/3 of the tube with protein solution, and you will boil it, heat it, and then what you get is the proteins coagulation occurring here, right? So if such kind of image given, so you can just detect that what is happening. Then next, coming to milky plasma. See, milky plasma doesn't mean that it's white plasma when you collect from the patient. No. Understand carefully what happens when you take blood sample from the patient. Blood is red in color, so obviously this will be red in color. So this also red, this also red. Let's say this is patient A, this is patient B. So next what we'll do, we will do centrifugation. Centrifugation we do for all the blood samples coming to lab. And then for checking whether the patient has milky plasma or not, then what we have to do, we have to keep this sample overnight at 4 degrees Celsius. And then in the morning, what we see this is patient A, this is patient B. So patient B has normal plasma. This is normal serum, right? Normal color. Then you can see patient A, this is milky. This is milky, right? Milky plasma. This is lipemic plasma, right? So you can see this is milky plasma. What it suggests, what it interprets that milky plasma means there are chylomicrons, right? Chylomicrons have floated to the top and have formed a creamy layer, okay? So in short, milky plasma means chylomicrons are increased in this patient, okay? Then a few images related to hyperlipoproteinemia only. So you can see here Palmer and tuberoeruptive xanthoma. Palmer, this is Palmer xanthoma, this is tuberoeruptive xanthoma. So these two xanthomas occur when remnants are increased. Remnants, by remnants I mean chylomicron remnant and VLDL remnant. VLDL remnant or IDL is one and the same thing, intermediate density lipoprotein. Then next image you can see tendon xanthoma. Tendon xanthoma means cholesterol is increased, right? Tendon xanthoma means cholesterol increased. Then let's see the shapes of osazones in case of reducing carbohydrates. Only reducing sugars will make crystals or osazones which can be seen under the microscope, okay? So first we will look at this one, needle shaped or broomstick appearance, right? Like this. In the microscope you can see this typical picture, needle shape or broomstick appearance. It occurs in case of glucose, mannose and fructose. They all will give this shape, okay? Then galactosazones, they will be rhombic plate like, okay, like this, rhombic plates. Then maltosazones. Maltose, in case of maltose, the crystals are of sunflower shape. So you can see this typical yellow and black color, exact similar to sunflower, you can see in the microscope. The shape is like this, okay? Then in case of lactose, it is hedgehog or powder puff, appearance, like this, right? Then let's see some of the organelles. Although I have separately taken one video on organelles and all the images are covered there also. So do watch that video also. So let's see. You can see this organelle is which one? This is outer membrane, inner membrane. Inner membrane is thrown into foldings or cristae, right? This central is the matrix. So you know this is mitochondria. So questions, they can give you the image of organelle and they can ask which pathway occur here. Okay, so you know that in mitochondria all the catabolic pathways occur. Which is anabolic, which is catabolic, that I have taken in the first video of formulas. So do watch that video. So all the catabolic pathways except glycolysis and glycogenolysis, because these are two catabolic pathways which occur in cytoplasm. Otherwise all catabolic pathways occur in mitochondria. Then vital pathways like TCA, ETC, they also occur in mitochondria. Then mitochondria has its own DNA. So that DNA can undergo replication, transcription and translation. And mitochondrial DNA is responsible for the formation of approximate 90% proteins of ETC. The remaining 80% will be coming from nuclear DNA only. Then activation of apoptosis occurs in mitochondria, and you should also learn that there are three pathways which occur both in mitochondria and cytoplasm, and these all three start from mitochondria. Okay, so the first step of these three pathways occur in mitochondria. Which are these three pathways? Urea cycle, heme synthesis and gluconeogenesis, right? Then next image, this is nucleus and the outer is nuclear envelope or nuclear membrane. So the continuation of nuclear envelope, is endoplasmic reticulum. When we have so many ribosomes attached, it is rough endoplasmic reticulum where protein synthesis occurs, proteins which are to be exported out of the cell, right? Then smooth endoplasmic reticulum where no ribosomes are attached. Here smooth endoplasmic reticulum function is in detoxification, synthesis of lipids and calcium release and sequestration, right? Then Golgi apparatus. Golgi apparatus will receive the proteins from endoplasmic reticulum. So mainly from rough endoplasmic reticulum. When vesicle is formed, this protein needs to be exported out of the cell. First it will be coming to Golgi apparatus, G for Golgi, G for gift packing. If we have to give something to other person, then we have to wrap it up very nicely, right? Gift cover. Okay, so when the cell has to give some protein outside of the cell, then the cell needs to wrap it up nicely. That wrapping up, gift packing occurs in Golgi apparatus. Okay, so this is Golgi apparatus. You should see the structure. This is receiving end of Golgi apparatus, and on the sides always vesicles are getting formed. These vesicles will go towards the plasma membrane, and this side of the Golgi apparatus is known as shipping side. The other side was receiving side. So from here the protein will be exported to plasma membrane. Then this is a free ribosome. You can see large subunit, mRNA, small subunit. Detail of this ribosome, how the work occurs here, is taken in the video of translation. Do watch that video, it is very, very important. So what happens in free ribosome? So earlier I have told you the ribosome on rough endoplasmic reticulum, okay, their function I have told you. Now the free ribosome function is to synthesize those proteins that are required inside the cell, in the cell metabolism. Okay, Then let's see some images in cell membrane transport, which is a very, very important topic of biochemistry. So you can see this image that on this side of the membrane, see this is a membrane, right? Lipid bilayer membrane. You can see there is a channel present here, okay, channel present here. This is the side of the membrane which is having high solute concentration, right? This side of the membrane has low solute concentration. The work of this transporter or channel is to take this solute from this side of the membrane to this side of the membrane. Okay. So, what is the mechanism followed by this protein or channel? It is ping pong mechanism. Ping pong means the protein has a different shape, right? It can change its structure, right? It can change its shape. You can see that image A and image B, okay? In image A, the opening is towards this side of the membrane when the solute is coming inside, okay? Then once this solute comes and gets attached here, once this solute comes and gets attached here, then the protein changes its conformation and now this side is closed and this side is open and the solute is released here, okay? So this is ping pong mechanism that is followed in case of glucose transporters, GLUTs, okay? So GLUTs is also very, very important topic. Do watch the video on GLUTs. Then let's see this diagram of intestinal cell, that which transporter is present at position X and Y. This is apical side of the cell, this is basolateral side of the cell, intestinal cell. So you can see here at the apical side the transporter is SGLT1. In case of intestinal cell we are talking here. So this is sodium dependent glucose transporter number 1 on the apical side, and at the basolateral side it is GLUT2 which is present in intestinal cell. Then also watch this image. This is enterocyte, again the intestinal cell. This is lumen side and this is blood, okay? So here at the lumen side of the cell, luminal side of the cell, there is SGLT1. Why SGLT1, which is sodium dependent transporter, so it is transporting sodium also, okay, or it is using sodium also. Then via this transporter both glucose and galactose can be transported. Then there is GLUT5 which is used for fructose transport here, okay. Then on the basolateral side GLUT2 is present which can transport glucose, galactose and fructose, okay. Then we also have sodium potassium ATPase pump at the basolateral membrane which transports 3 sodium out of the cell and 2 potassium inside the cell. Then this is renal cell, renal tubular cell. So this side is blood, this side is lumen, and here we have SGLT1 or 2, 1/2 is written. So either SGLT1 or 2 can be present in kidneys. And then on the basolateral side we have sodium potassium ATPase pump and we have GLUT2. Then we can have a look at the pictures of phagocytosis, which is cell eating. Some solid particle when taken inside the cell via the cytosis of the plasma membrane, that is called phagocytosis. Then pinocytosis means cell drinking. So some solute or some liquid if taken inside the cell, then it is called pinocytosis. Then we have receptor mediated endocytosis. In this case there is a pit in the membrane, which is coated by protein, by a special protein, that is clathrin. This is a special kind of endocytosis known as receptor mediated endocytosis. One example of receptor mediated endocytosis is LDL receptors. LDL receptors present on liver cells, hepatocytes, and they will uptake LDL from blood via receptor mediated endocytosis. Then uniport, symport and antiport. See, uniport means only in one direction, right? So in one direction only one substance is taken from one side of the membrane to other side, whether it is going inside the cell or outside the cell. Okay, then the other two are co-transport methods. Co-transport means two molecules are taken together. In symport, both the molecules are going in same direction, okay, and in antiport, the two molecules are going in opposite direction. For example, this blue one is going this side, the purple one is going this side. Okay, then see the proteins in the lipid bilayer membrane. This protein is covering the entire length of the membrane, known as integral membrane protein. Example, sodium potassium ATPase pump, right? Then we have some peripheral membrane proteins, so towards the outside. So this is outside of the cell, you can say this is inside of the cell. Okay, then there are some lipid anchored proteins. Mostly the proteins are G proteins. They are anchored in the cell membrane via a lipid, and that lipid is phosphatidyl inositol, right? These are known as lipid anchored proteins. Then let's see the diagram of sodium potassium ATPase pump. It has two subunits. I told you it is an integral membrane protein. So it has two subunits, alpha and beta. Alpha is the main subunit or main part. Beta is just for anchoring this protein in the membrane, right? Then this alpha protein, alpha portion has sodium binding site, potassium binding site, phosphorylation site, ATP binding sites. Most of the binding sites are on alpha subunit, and beta subunit has glycosylation sites on it. Glycosylation sites towards the outside of the membrane, right? And so this protein, we have seen it is a dimer made up of alpha and beta subunit. Then let's see some images required for your clinical cases, some histo images or patient images here, right? So Gaucher's disease. Gaucher's disease is most common lysosomal storage disease, and the peculiar feature here is crumpled tissue paper appearance of this Gaucher's cell. This cell you can see resembles this, it resembles this crumpled tissue paper, right? So this cell. So let's do little detail of this Gaucher's disease. In Gaucher's disease, patient has hepatosplenomegaly, bony pain and pathological fracture occurs. There's pancytopenia, thrombocytopenia. So patient has easy bruising. Then X-ray of long bones. X-ray of long bones shows Erlenmeyer flask deformity. See, this is Erlenmeyer flask, and this bone, long bones, resemble this Erlenmeyer flask because they are the ends of long bones. Ends of long bones are expanding and lytic, right? Because here this Gaucher's disease is a sphingolipidosis. Sphingolipids are getting accumulated. Okay. Now this Gaucher's disease has no mental retardation and no cherry red spots. Otherwise, most of the sphingolipidosis have cherry red spots, right? Then this is the cherry red spot, which is macular cherry red spot found in sphingolipidosis. but all sphingolipidosis have cherry red spot except Gaucher's, I told you already, and one more, that is Fabry's disease. Okay, it is Fabry's disease. Then angiokeratoma. Angiokeratoma are benign cutaneous lesions of capillaries resulting in small marks, red to blue color, and they are characterized by hyperkeratosis. So, this angiokeratoma is found in which sphingolipidosis? It is found in Fabry's disease and GM1 gangliosidosis, right? Then you can also see this picture of angiokeratoma, so you are able to recognize when image based question is asked. So, this is angiokeratoma. Then sphingolipidosis resembling rheumatoid arthritis is Farber's disease. It is not Fabry, it is Farber's disease, right? Patient has joint deformities, arthritis kind of picture, similarly like rheumatoid arthritis, and this is Farber's disease, okay? Then you can see this picture of adrenals where there is calcification of adrenals, that is Wolman's disease, which is a lysosomal storage disease but not a sphingolipidosis. This is also a disease where lipids are getting accumulated. The enzyme deficient is lysosomal acid lipase. Some of the clinical features are watery green diarrhea, vomiting, failure to thrive, hepatosplenomegaly, and Wolman's disease is also known as cholesterol ester storage disease, right? Then you can see some cataracts. Snowflake cataract. Snowflake cataract is found in diabetes, right? Due to the excess of alcohol sorbitol. Then oil drop cataract is found in galactosemia, and this is because of excess of alcohol, galactitol or dulcitol. Then this is sunflower cataract, which occurs in Wilson's disease. Then see this image. Can you see this? Can you tell what is this? Bleeding from here, bleeding from here. Can you see this is self-mutilation by the patient? It is a neuropsychiatric symptom which occurs in Lesch-Nyhan syndrome. Lesch-Nyhan syndrome, where there is defect in enzyme HGPRT, hypoxanthine guanine phosphoribosyl transferase, and also these patients have gout. Right? Then alkaptonuria, where urine turns black in color on long standing. It is due to oxidation of homogentisic acid. The theory details we have already covered. You can just see the pictures. Here there is discoloration of ear cartilage, bluish discoloration of sclera, spots, blackish spots on the eyes. Then there is damage to the calcification of the intervertebral disc, and because of this thing, patients have chronic back pain. Then there is bluish black discoloration of skin, right? So all these pictures can be used for giving you a clinical question on alkaptonuria. Then let's see albinism, where patients have milky white skin, white hair, that is because of tyrosinase deficiency. This enzyme helps in formation of melanin pigment. Okay, so completely white skin and white hair. But if it is patchy skin, that is vitiligo. Patchy skin will be vitiligo, okay, and completely white will be albinism. And there is one more disease where patient has hypopigmentation, which is that, yes, that is phenylketonuria. So in phenylketonuria, amino acid phenylalanine cannot form tyrosine, and tyrosine cannot form the pigment melanin, right? So also very important thing that these patients have, in addition to hyperpigmentation, the main feature is mental retardation, right? So this is an adult untreated case of phenylketonuria. So you can clearly see in the picture that this patient looks mentally retarded, right? And in the above pictures, you can see the hypopigmentation, and usually these patients have bluish sclera. The next is homocystinuria. In homocystinuria, patients have dislocated lens, pectus carinatum, genu valgum, short stature, lens dislocation, deformity of chest, that is pectus carinatum, and osteoporosis. And also in homocystinuria, patients can have myocardial infarction, stroke, and vascular diseases, right? Then mucopolysaccharidosis. Mucopolysaccharidosis, topic already covered in detail in a separate video in carbohydrates, that is mucopolysaccharidosis only. Okay, here we can just see the images. Hurler disease, which is type 1 mucopolysaccharidosis, Hunter disease, which is type 2. There is corneal clouding in Hurler, but clear vision in Hunter disease. And the patients have coarse facial features, mucinous discharge coming out always, right? And bullet shaped middle phalanges, right? Then also in mucopolysaccharidosis, some more X-ray findings. Bullet shaped phalanges we have done, broad and proximally pointed short metacarpals, right? Then in mucopolysaccharidosis, we have Rayleigh body inclusions. Rayleigh body inclusions are coarse purple granules, large coarse purple granules in neutrophils, right? So mainly they are found in type 1, that is Hurler disease. Then see this image. The child has upslanting eyes, high forehead, skin folds along the nasal borders. This picture is typical picture of Zellweger syndrome, which is the most severe paroxysmal biogenesis disorder where there are empty peroxisomes. The enzymes are not there in peroxisomes, also known as ghost peroxisomes. Okay. Theory part I have already covered in the videos. Then von Gog's disease, which is type 1 glycogen storage disease, also the most common glycogen storage disease in children. You can see here doll-like faces because of hyperlipidemia, collection of fat. Then patients have hepatomegaly. So you can see the distended abdomen of this child. Patients have lactic acidosis, hyperuricemia, and severe hypoglycemia. Then let's do some images in vitamins and minerals. Vitamin B2 deficiency, riboflavinosis. Patient has magenta tongue, or you can say glossitis, because of the inflammation it is magenta in color. Then corneal vascularization or pink eye. Then dermatitis, chelosis, inflammation of corners of mouth, right? So these all occur in riboflavin deficiency, known as riboflavinosis. Then you can have a look at Kiesel's necklace, which is frequently asked topic in image-based question also. So this is side of neck shown. This is front of neck shown of other patient. So this is dermatitis occurring in pellagra. Pellagra is vitamin B3 deficiency. Okay, so there is photosensitive dermatitis in these patients like this, Casel's necklace or on the face, or all sun exposed parts. So feet, hands, right? So this is photosensitive dermatitis. Then coming to vitamin A deficiency, where we have Bitot spots. These are superficial deposition of keratin in the conjunctiva. You can see superficial deposition of keratin in the conjunctiva, and a typical feature is follicular hyperkeratosis. Then Cation's disease, which is selenium deficiency. It initially was found in a province of China known as Cation. That's why the name Cation's disease. Mainly it affects children and women of childbearing age. Patients have cardiomyopathy, muscle weakness, can have muscular dystrophies. These patients are having inability to gain weight. Hypothyroidism can occur, hypertension, eczema, arthritis, and even cancer can occur. Even cancer can occur. If you remember, selenium is required for enzyme glutathione peroxidase, and that is helping in destroying free radicals, right? So if free radicals are not destroyed, they can damage many genes, and it can lead to cancer. So selenium poor soil correlates with high incidence of cancer, right? Then coming to this, see this brown, golden brown pigmented ring in the Descemet's membrane of cornea. This occurs in Wilson's disease, which is the condition of copper excess in body, right? So this is due to deposition of copper in the Descemet membrane of cornea. Okay. Then Menkes disease, which is a condition of copper deficiency in body. These patients, you can see all these different images here. They have light hair color, light skin color, and mental retardation. MR is mental retardation. Why again hypopigmentation is occurring in Menkes disease also? Because copper is required for enzyme tyrosinase. Tyrosinase is an oxidase, and all oxidases require copper, and tyrosinase enzyme makes melanin, okay, the pigment melanin. Then this is the structure of glucose. Many questions are solved indirectly if you know the structure of glucose. So see, it's a 6 carbon sugar, it's aldehyde sugar. So first carbon is aldehyde CHO, and last is CH2OH, the terminal carbon is CH2OH, and on all the remaining carbons it's mainly H on left hand side, OH on right hand side, right? H on left hand side, OH on right hand side. But at C3, you have to learn at C3, OH is on left hand side. Okay. So learn by heart the structure of glucose. Then can you tell me which is this structure of protein? Primary, secondary, tertiary. So this is all the amino acid sequence is given. So this is primary structure of protein. Then this one is alpha helix. Alpha helix. So this is secondary structure of protein. Then this is 4-folded polypeptides. This is quaternary structure of protein. If only single polypeptide chain given, that will be single fully folded polypeptide chain, that will be tertiary structure of protein. Then this is beta bend or beta turn. Question asked which amino acid is present mostly at beta bend or turn. So at beta bend, glycine and proline. Only these two amino acids can be found in beta bend or turn. And usually the number of amino acids present here are 4. Okay. Which is this protein where many glycine and protein residues are there. So glycine, right? Many prolines. What is this? Which protein? This is collagen. You remember in collagen, every third amino acid, in the primary structure of collagen, every third amino acid is glycine. And the primary structure is glycine XY repeated n number of times. Okay, like this we write. So every third amino acid is glycine. Then, which is this amino acid? This amino acid, can you see this is carbon? This is the central carbon. On one side carboxy, on one side amino, and the fourth side is, so there is one H on one side, the fourth side is R. Okay, R means the side chain. And you can see here the side chain is attached to the primary amino group. So this thing occurs in case of proline, right? That's why proline is secondary amino acid, not a primary amino acid, right? So this is proline. Then glucose residues here in glycogen. So the center portion is glycogenin, and that is the central portion, and there are so many glucose residues tightly packed here. So which bond, question asked, which bond here at point A, B and C? At point A, it is linear structure. You can see it is linear structure. So this is alpha 1,4 bond. Point C also is in the linear structure, that is also alpha 1,4 bond. But point B, at a place where branch is starting, branch point is alpha 1,6. Okay. Then some slides of chromatography and electrophoresis. So you can see this, which kind of chromatography is this? There are larger and bigger molecules going down in this column. So one, I can say this is column chromatography. But which type of column chromatography? This is gel filtration. You can see as proteins are going down, this bigger protein is going quickly downside, right? And smaller protein is entering the gel and coming slowly. Okay, so this occurs in gel filtration or size exclusion chromatography, right? Then what is this chromatography? When this is the column where positive ion is attached in the column, and this is mixture of positive negative amino acids going down. So what is happening? Negative amino acid will be attached to the column and positive amino acids are eluted out. So this chromatography is ion exchange chromatography, right? Then the structure of some amino acids. Let's see, you should by heart learn all the structures of amino acids, or at least if the structure is there in front of you, you should be able to recognize which amino acid is it, right? Like this is valine, leucine, isoleucine. They are branch chain amino acids. So this is the common structure, right? And there is this structure, there is a V in case of valine. Can you see? This is a V, carbon here, carbon here, carbon here. So V in case of valine. Then leucine. In case of leucine, you can see L. So one carbon, other carbon, carbon here, carbon here. So one L kind of structure here, right? Then isoleucine and leucine have same number of carbons, just that the position of carbon here is, the position of methyl group is here, right? Just a difference in the position in leucine and isoleucine, otherwise number of carbons are same. Okay. Then phenylalanine, having this phenylalanine ring and a carbon other than the basic structure. Okay. In tyrosine, just one OH added in the structure of phenylalanine. Then tryptophan, there are two rings known as indole ring or nucleus, right? Then this is the structure of histidine. Histidine having side chain, one carbon and a ring in the side chain, ring having two nitrogens in the ring, that is imidazole ring. Because of this imidazole group, histidine has good buffering capacity. Then structure of aspartate, glutamate, asparagine, glutamine, right? In asparagine and glutamine, there is NH2 group added, right? There are 4 carbons in aspartate and asparagine, 5 carbons in glutamate and glutamine. Okay, so you should be able to recognize the structure of amino acids if given in image based question. Then lysine has after the basic structure, it has 4 carbons and 1 amino group. Okay, arginine has 3 carbons, and then this group known as guanidino group. Guanidino group has center carbon with 3 nitrogens. All 3 sides have nitrogens, right? On one side there is a double bond with nitrogen, okay, because we have to complete the valency of carbon. Then histidine, I've already told you. Then serine. In serine, after the basic structure, there is CH2OH, this one carbon with OH. In threonine, there are 2 carbons and 1 OH. Okay, you just add 1 methyl group in serine, it becomes threonine. Then tyrosine, we have already done. Then nitrogenous base, purine pyrimidine structures. So first let's do purine. 2 rings means purine. Okay, when 2 rings and amino group present here at the top, then it is adenine. When 2 rings and amino group present here on the side and here 1 oxygen present, that is guanine. Okay, pyrimidine 1 ring. Cytosine means 1 amino group present, right? Uracil, when no amino, no methyl present. Thymine, thigh is matching with methyl. Okay, so methyl group present here, that is thymine. Okay, so let's revise. If 1 ring and amino present, that is cytosine. 2 rings amino present at the top position, this position, always look for this position for amino. 2 rings amino present, that is adenine. Now 2 rings O present here, that is guanine. 1 ring methyl present, thymine. 1 ring no amino, no methyl, that is uracil. Okay, so now tell me, what is the structure? This is a nitrogenous base, this is a purine, 2 rings are there, amino group present here, so this is adenine nitrogenous base. But this adenine nitrogenous base is also attached to this sugar. When adenine is attached to sugar, it becomes a nucleoside. In case of adenine, the nucleoside is known as adenosine. So this is adenosine. Okay, do watch the video on nucleotides also. So there I have covered all the things, basics there, right? Then what is this image? This is a karyotype where all the 23 pair of chromosomes are shown, and this karyotype is best technique for detecting monosomy and trisomy, right? Then what is this technique? This is microarray, or you can say chip, gene chip, right? What is here, that this is patient's DNA taken, and this is fluorescent complementary DNA, which is the probe is labeled. And what we do, that on a chip, on a small material, we can have so many probes. So first probe is placed here, then minimum amount of sample added, and multiple mutations can be detected by this microarray or gene chip. This kind of image also if comes, so you can easily tell this is microarray. Okay, then one of the enzyme graph, then the graph between substrate concentration and velocity. The rectangular hyperbola graph is for simple enzymes, which are most of the enzymes. The sigmoidal graph is for allostric or regulatory enzymes. Okay, do watch the video on enzyme kinetics where I have covered all other enzyme graphs also. So this is it for this video. Thanks for watching and best wishes to you.